Hello everyone. So I first would like to thank the organizers for their kind invitation. So I would like to tell you about our work on the photoreactions of fluorescent proteins, which includes both time-result spectroscopy studies and some methodological developments in bioimaging. So you have already heard of fluorescent proteins during the previous talk, but so there are a family of proteins that are related to the green fluorescent protein from the jellyfish Aequorea victoria here. And here you can see their chromophore, and it has the particularity that it is encoded in the sequence of the protein. It forms by spontaneous cyclization of these 3 amino acids here after protein folding. And the protein adopts a beta-barrel structure, which protects the fluorophore from the environment and also prevents it from twisting. And this is how we obtain this bright fluorescence. So the fluorescence of fluorescent proteins is genetically encoded, and this is what makes them so successful as fluorescent probes for bioimaging. Most FPs that are used for imaging are actually artificial proteins obtained by genetic engineering of the natural FPs. And genetic engineering made it possible to build a whole toolbox of FPs with different colors and properties that biologists know how to fuse to their proteins of interest to make them fluorescent and observe them under the microscope. However, fluorescent proteins are not just fluorescent. They also have a very rich photochemistry with many photoreactions reported. Some of these photoreactions affect the chromophore, others affect the protein, and still others affect the two. And their mechanisms are only partially understood, although the photochemistry of FPs has important implications for their use in imaging. The photochemistry of FPs is a drawback when it leads to unwanted fluorescence losses, such as photobleaching or single molecule blinking. On the other hand, it is also at the origin of several advanced imaging methods, such as super-resolution imaging. These methods often use special phototransformable FPs that can be interconverted by light between several states with distinct spectroscopic properties. So in my group, we use standard transient absorption spectroscopy to study the mechanisms of the photoreactions of FPs. And our main tool is a homemade, setup for broadband femtosecond transient absorption spectroscopy that is pumped by an amplified femtosecond titan sapphire chain from spectrophysics. With this pump probe setup, we can access delays up to 3 nanoseconds, and when we need to probe slower reactions, we also use real-time transient absorption spectroscopy, either in collaboration. At the moment, I have a collaboration with Pavel Müller at CEA Saclay, or we are also developing our own setup at ENS. So this here is an example of a transient absorption spectra that we measured using our femtosecond setup on a fluorescent protein called Dreiklang Y203F. These spectra are the difference between the spectrum of the sample excited by the pump and the spectrum of the sample in the ground state. Therefore, they comprise both positive and negative contributions. There are actually 3 types of contributions. Negative contributions from ground state bleaching and stimulated emission that can be identified by comparison with the steady state absorption and fluorescence spectra, which are shown here. And there are also positive contributions due to the absorption of the excited states or of the photoproducts formed by the pump. Then the spectra evolve with time as the photoreaction progresses. And here, for instance, we see a decrease in the stimulated emission of the protonated chromophore here, and a concomitant increase in the stimulated emission of the deprotonated chromophore around 510 nanometer. This spectroscopic signature is well known to FP specialists. It is the signature of excited state deprotonation of the chromophore at the level of this phenol group here. We've spent quite a lot of time in the group studying the mechanisms of reversibly switchable fluorescent proteins, RSFPs. RSFPs are a subcategory of phototransformable FPs that can be reversibly photo-switched between a fluorescent state and a non-fluorescent state using two wavelengths of light, lambda 1, lambda 2. And a number of these RSFPs have been studied by static X-ray crystallography, which reveals that there are two types of photo-switching mechanisms. Cis-trans isomerization of the chromophore coupled with proton transfer is in the vast majority of RSFPs. And in one protein only, Dreiklang, the reversible hydration of the imidazolinone ring of the chromophore by a nearby water molecule, which is shown here. The details of these photoreactions have been studied by several groups using time-resolved spectroscopy as well as time-resolved crystallography, and I will now show you our contributions to this work. So I'll start with Dreiklang. In a first experimental study using femtosecond transient absorption spectroscopy, that was in 2017, we observed an ultra-fast excited state deprotonation of the chromophore with an apparent time constant of 100 femtoseconds. A close look at the chromophore environment in the crystal structure of Dreiklang led us to propose that the proton lost by the chromophore here is transferred to this histidine 145. In the same paper, we further reported that chromophore hydration is not completed at 10 nanoseconds, since the spectrum measured at this time scale does not show the UV absorption expected for the off state. Instead, it shows an absorption at 450 nanometer. That is a characteristic of an intermediate that we named X. Dreiklang was then studied theoretically by the Krylov and Nemukin groups, and the theoreticians did not observe ESPT in their simulations. Instead, they proposed that photohydration is initiated by electron transfer from this tyrosine 203 here in gray to the chromophore. And they further proposed a structure for intermediate X. They proposed that X is a complex of the cation radical of the chromophore with the anion radical of tyrosine 203 and the water molecule involved in hydration. So following on from this theoretical work, we have re-examined experimentally Dreiklang in order to clarify the role of proton and electron transfer and to characterize the dynamics on timescales longer than 10 nanoseconds. To assess the role of proton and electron transfer, we mutated histidine 145, which is the putative proton acceptor, to non-protonatable tyrosine, and tyrosine 203, which is the putative electron donor, to non-oxidizable phenylalanine. Under 410 nanometer irradiation, we found that the H145Y mutant photohydrates like Dreiklang, while the Y203F mutant has lost the ability to photohydrate. This is a strong indication that tyrosine 203 plays an important role in photohydration, but histidine 145 does not. To determine the timescale of hydration, we then measured the kinetics of formation of the hydrated off state in the UV by nanosecond transient absorption spectroscopy, and we found a characteristic time of 30 nanoseconds in Dreiklang, which also corresponds to the decay time of intermediate X. Ground state bleaching, shown here, is stable at this time scale, which suggests that the off state is formed directly from X with 100% yield. We also used the UV transient absorption signals to determine photohydration yields, and we found, so we used bipyridine ruthenium as an actinometer for this measurement, and we found a value of about 0.5% for both Dreiklang and H145Y, so very low quantum yield. Analysis of the femtopicosecond transient absorption spectra led us to conclude that ESPT is not involved in photohydration, but that it is a parallel pathway, and that histidine 145 is indeed the proton acceptor. So the signature of ESPT here is indeed conserved in Y203F, where it is even more pronounced than in Dreiklang, but it is suppressed in H145Y. So we have one mutant, Y203F, that doesn't photohydrate but shows ESPT, and another mutant, H145Y, that photohydrates but doesn't show ESPT. This is a strong indication that the two processes are independent. In addition, the suppression of ESPT in H145Y suggests that histidine 145 plays an essential role, likely as the proton acceptor. For Dreiklang and H145Y, but not Y203F, the subsequent dynamics involve a step that is characterized by significant changes in the blue part of the spectrum only here. I'll come back to this step in the next slides. And all three samples then show a decay of all bands due to the decay of the excited state. Finally, at the 10 to nanosecond scale, we see small residual signals due to ground state photoproducts. So the fluorescence excitation spectrum of Dreiklang enabled us to estimate the ESPT yield at only 5%. And ground state bleaching is stable at the ESPT scale, which rules out a massive return of the excited population to the ground state. So the low ESPT yield indicates that something else is going on in parallel with 95% yield, and that also dominates the short component, the 100 femtosecond component. The decay-associated different spectra provide more information on this process. Of particular interest is H145Y here, in which ESPT is suppressed. The DADS of the first component, 145 femtoseconds, is dominated by a large negative band here at 470 nanometer that we assign to the rising absorption of a dark state. In contrast, the DADS of the second component, 920 femtoseconds, features a positive band at this place, which indicates the decay of the dark state. A very similar signature is observed in the two first DADS of Dreiklang, but not in those of Y203F. Since Y203F was designed to disfavor electron transfer from tyrosine 203 to the chromophore, and since it does not photohydrate, it is very tempting to assign the dark state of Dreiklang and H145Y to the charge transfer state identified by the theoreticians, and to assume as they do that it is involved as an intermediate in photohydration. So we propose, in line with the theoreticians, that the main excited state deactivation pathway is the formation of this charge transfer state. However, since the photohydration yield is only 0.5%, we further propose that the CT state decays massively to the ground state in less than a picosecond, with only a small part of it evolving to the off state. So the light-induced dynamics of Dreiklang are complex and multiscale, and they involve non-predictive pathways in addition to photohydration. As far as photohydration is concerned, our data are compatible with the theoretician's proposal that it starts with the ultra-fast formation of a charge transfer state from tyrosine 203 to the chromophore. However, we showed that this CT state decays predominantly and very fast to the on state, and that the yield of formation of intermediate X and of the off state is only 0.5%. In addition, hydration is quite fast, as it occurs in 30 nanoseconds. We've also studied the much more common photo switching by cis-trans isomerization and proton transfer on the example of DROMPA, but I won't go into as much details here due to the lack of time and also because this is older work. Our main contribution was here that we were able to show that the main photo switching intermediate at 300 picoseconds is a cis-phenol form based both on its absorption spectrum and on its anisotropy. So this led us to conclude that cis-trans isomerization occurs first in the excited state at the picosecond time scale. And we were also able to measure the proton transfer step here. And we found that it occurs in the ground state at the microsecond time scale. So now I would like to tell you a little bit about recent work in the field of bioimaging. Beyond the fundamental interest of time result spectroscopy studies, we are indeed increasingly seeking to exploit our knowledge of FP transient states to propose improved imaging techniques. And we have focused more particularly on two major issues in fluorescence imaging. One is multiplexing, that is how to increase the number of fluorophores that can be imaged and distinguished at the same time. And the second one is the control of the photobleaching of probes. So photobleaching of probes is the gradual loss of fluorescence of probes during imaging experiments. So our work, our fundamental work, and that of other groups, have shown that the photo-switching cycle of Drompa-type grid RSFPs comprises mainly 4 steps. So when you start from the on state, the chromophore first isomerizes, and then it protonates. This leads to the off state. And then return to the on state is also achieved by isomerization followed by proton transfer. The timescales of isomerization and proton transfer are very different. Isomerization takes place at the picosecond scale, while proton transfer takes place at the micromillisecond scale, depending on the protein. And another key point is that the characteristic times of these 4 steps are specific of each RSFP. So this gave us the idea to exploit the photo-switching dynamics of RSFPs as a contrast factor in biological imaging, so to discriminate between the different RSFPs that have the same color of emission. To access the dynamics of the 4 steps under the microscope, we monitor the fluorescence of the samples while submitting them to the following illumination sequence. First, a low intensity at lambda 1 and a low intensity at lambda 2. At low intensity, on-off and off-on switching are kinetically limited by the light-dependent step, so by cis-trans photoisomerization of the chromophore. And therefore, the characteristic times that we measure here are proportional to the photoisomerization cross-sections. Then we continue with high intensity at lambda 1, followed by high intensity at lambda 2. At high intensity, photoisomerization becomes faster than proton transfer, and switching on-off and off-on switching are kinetically limited by the proton transfer steps. Therefore, we access here directly to the characteristic times of proton transfer. So this method that we named lightning enabled us to discriminate 9 RSFPs, 9 green RSFPs expressed in bacteria using a single green spectral channel. By exploiting several spectral channels, it could be used to discriminate tens of fluorophores. So lightning is a possible answer to the problem of multiplexing in fluorescence imaging. We have more recently turned our attention to another major issue in imaging, the photobleaching of fluorescent probes. The mechanisms of photobleaching are incompletely understood, but it is generally admitted that they involve the fluorophore triplet state as an intermediate. So T1 is formed from S1 by intersystem crossing, and it then reacts with oxygen to form reactive oxygen species that degrade the fluorophore by oxidizing it. So this leads to photobleaching of the probe. ROS are in addition toxic to living samples, so that photobleaching is generally accompanied by phototoxicity. It has long been known by spectroscopists that the triplet states of many molecules, so that the relaxation of the triplet states of many molecules can be accelerated by light through a photophysical process called reverse intersystem crossing, RISC. RISC is achieved by exciting the fluorophore from its lowest triplet state T1 to a higher triplet state Tn, from which it transitions back to singlet excited states and then decays to the ground state. The risk-inducing wavelengths, lambda 2 here, must coincide with the triplet-triplet transition. It turns out that the triplet absorption spectrum of EGFP has recently been published and that it shows a maximum at 900 nanometer. We therefore wondered whether it would be possible to reduce the photobleaching of EGFP under visible light excitation by co-illuminating it at 900 nanometer. To verify this, we immobilized purified EGFP in a polyacrylamide gel, and we illuminated it at 470 nanometer over the whole field of view with co-illumination at 900 nanometer only inside the red circle. After a few minutes, only the co-illuminated region was still visible. We extracted the photobleaching kinetics in the presence and absence of co-illumination, and they show that under optimized conditions, co-illumination increases the number of photons emitted by EGFP by a factor of 3.5. We also took advantage of the tunability of our infrared source to measure the action spectrum of near-Earth light, and we found that in the linear regime, it corresponds perfectly to the published triplet absorption spectrum of EGFP, which proves that reduced photobleaching is indeed due to risk. To specify the illumination conditions under which the effect occurs, we further studied its intensity dependence, and we found that the effect increases with near light intensity up to about 1 kilowatt per square centimeter, where it reaches a plateau, and that it decreases for visible light intensities higher than about 100 watts per square centimeter. This means that risk should be particularly effective to reduce photobleaching in wide-field fluorescence microscopy and other fluorescence microscopy techniques that use low to moderate excitation intensity. We checked that the method works as well on live cells. So these are EGFP labeled HeLa cells, and we found that the effect of near light is similar as in vitro. We also compared the effectiveness of near light with that of the anti-bleaching cell culture medium DMEM GFP2, which is currently the main available solution to reduce the photobleaching of FPs in live mammalian cells. And we found that near light is more effective than DMEM GFP2, and that it is possible to obtain an even higher six-fold improvement by combining both strategies. Finally, we reasoned that by quenching the triplet state, near light should also reduce the production of ROS and hence phototoxicity. Cell proliferation is a well-established criterion to assess phototoxicity. We therefore followed the growth of E. coli microcolonies labeled with a yellow FP YPET, so in the presence and absence of near light. So we found that bacteria illuminated only at 517 nanometer, which is the excitation wavelength of YPET, show a very slow growth rate due to the phototoxicity of this visible light, while dual illumination at 517 plus 885 nanometer restores a normal growth rate. So we conclude that near light has indeed a protective effect against phototoxicity due to the visible excitation of FPs. So this brings me to the conclusions. I have touched on quite a lot of different topics during my talk, but I hope that I have convinced you that the photochemistry of FPs is very rich. It involves a wide variety of mechanisms and transient states with lifetimes that cover all time scales. Time result spectroscopy is a valuable tool for characterizing it. We have used it successfully to elucidate both types of RSFP photo-switching mechanisms, and we plan also to use it in the near future to characterize triplet states and risk at a fundamental level. And finally, knowledge of FP transient states can be exploited to improve biological imaging. The lightning method that we have developed uses the photo-switching dynamics to distinguish between RSFPs of the same color, while light-induced risk reduces photobleaching and phototoxicity. I would like to conclude by thanking all the people who were involved in the work I have presented today. A very important person at ENS for spectroscopy is Fabien Lacomba, our engineer. The work on Dreiklang was mostly performed by my student Émilie Renoir, that on Drompa by Dhirendra Yadav, and the work on RISC by Lucie Ludvíková and Emma Simon. The lighting method was developed in close collaboration with his two colleagues, Thomas Lesseau and Ludovic Julien. These are my collaborators for nanosecond transient absorption spectroscopy, and the people in the last column, my collaborators on the RISC project. So I also thank funders. And I would like to take the opportunity to mention that I have 2 years of funding available from early 2026 for a postdoc or a research engineer to extend the RISC work to other fluorophores. The position will be at ENS in the center of Paris, so if you're interested, please do not hesitate to contact me for more details. And thank you all for your attention.